हेलो शायद में। हेलो 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 शायद में। आवाज़ आ रही है अब? ठीक।
So in the nature, so long as there is no external influence which has been provided, there is a proper balance which is there. That will be there.
How profitably biodiesel can be harvested from it? Because
There is one good one which is there in Bihar where open production of algae is being worked out. There is one more with marine algae which is done in Gujarat, the shores of Gujarat, right? But with algae, the problem which is coming up is the high density cell culture. So you need algae in good amounts which can be easily removed from the system and then be taken out. Because when you are dealing with large amounts, you can't look at the process of centrifugation. Right. So the biggest challenge with algal growth is number one, robust in nature, so that you can go with large scale production in say open tanks, otherwise photobioreactor. Number two challenge is increasing the cell density. Number three challenge is managing the cell density with the lipid accumulation within it. So these are some two, three very prominent hurdles which are causing the issue. It can be profitable because compared to plants, like plants, you need a full growing season to harvest the seeds, then crush it into oil. Here, you can like manage to get a productivity. Say you can optimize it like in 12 days, one cycle is done. So where 12 days one cycle is done, it can go like in a month you can get two cycles. Compared to a plant, automatically, hypothetically speaking, the algal harvest, the algal yield can be much more. It is since it will be lighter, you can process it there or you can dry it and send it. Those things are also present. But the whole problem is increasing the density and matching it with the lipid accumulation within it. That is the point.
Any other question?
And what is your thought? Which aquatic ecosystem, like the coal, like you have taken from the Rajasthan, and there are some water bodies in the Ladakh. So generally, the coal system, the oil, the body needs oil actually. Every organism's body. So, what is your thought? Which ecosystem is likely have more diversity for the biofuel?
Sir, if we go to the colder ecosystems like Ladakh, etc., the psychrophilic or the psychrotolerant ones will be more found over there. But with them, the PUFA content will be greater. Because as the temperature goes down, the cell stability or other factors, you will not have the saturated fatty acids will be a little less. And if you take a look at the psychrophilic diversity per se, if you talk about the psychrophilic diversity, sir, the microalgal diversity, I'm talking about the eukaryotic algae over here, that diversity will decrease, sir. That will decrease because if you are going as it will progressively start to become colder and it keeps on increasing, the psychrophiles which are generally there, the microalgae can be there. I'm not saying they will be totally absent. Within a certain range you will get it, but yes, the diversity definitely will be much lower. Your diversity will be much better and much higher if you are going to come down into the subtropical, semi-tropical, that region, this whole part which
There
is
are
there.
few reports from Rajasthan also where they have, from the marble mining, they have got some certain, microalgae, and that is, they are
Yes,
trying to
that is the point, sir.
so
We need to increase the range of isolation over here.
isolation. This
There.
all depends, whether it is going to click for you or not.
Because
you,
the
you can get
cell
from
count is so
anywhere.
low
You can
that
get
getting
from
it,
anywhere.
yeah, that is.
Thank you, Dr. Patil. Yeah, one more question. Well,
Yes.
somebody will answer. No. Okay, thank you. Oh, yes, he wants to.
Yes, please.
how efficient is this biofuel, and can it replace other fuels?
You can perhaps not replace the conventional fuel with one source.
At least
At
10%
least,
or 15% you can
yes.
add.
It is a fraction which can be covered. But the conventional fuels, it is as of now, on the basis of what existing science technology we have, it can't be 100% replaced
So
because the usage is too big, too big.
if you go to Sweden, I have visited there. So there, most of the government buses they are running on this biofuel. This is a combination basically. You
Combination.
cannot have just only for biofuel. Right? You can add
Reduce from some part,
10
inject in some other
15%.
part.
That you can add.
We have this E20 now, E20 petrol, like ethanol.
Yeah.
So blends,
This that
yes,
is already
that is
blended.
there.
That the government is.
Hmm. Even biodiesel blends are being accepted, and now it is also there.
What is the cutoff of lipid percentage you put your biofuel?
I did not per se identify any lipid percentage cutoff. I just looked at those which showed me a greater value. So if the difference between the two yields was say like 50 units or 40 units for the temporarily for the current study, I didn't focus on them at all. But these isolates are still there with me. So if I have to go back again, recheck, and then identify or quantify on the basis of other parameters, I can do that. So right now, I simply focused on the neutral lipid yield which I got. That was my point.
Thank you, Dr. Kakule,
Thank
for
you,
a
sir.
nice and exciting lecture.
Thank you for being so patient.
Yeah. And,
Answer.
next speaker will be Dr. Dhan Prakash Wankhede. And Dr. Wankhede is a senior scientist of plant genetics in the Division of Genomic Resources in ICR NBPGR, New Delhi. He has 14 years of research experience in the capacity of postdoctoral researcher at NIPGR scientist, senior scientist, ICR, NBPGR, New Delhi. This area of expertise include genome analysis, bioinformatics, association genetics, and structural and functional genomics. He has published more than 60 papers in our area. So one good thing that like I want to add beside this that he was my student as well. So thank you. So it's really honor, that my student is speaking.
It is an honor to me. All right. So good afternoon. It is a delight to be here at the institute. This is the prestigious Institute of Higher Studies, and double delight that, as Dr. Praveen Verma sir told, that he was my teacher at NIPJ during my PhD, and I have a high regard for him as a scientist and as a teacher. So, the topic of my presentation is germplasm genomics, and we have taken example of linseed, that is a flaxseed. So, you must have seen this flaxseed or the linseed in the supermarkets, but you may not have seen the plants. So, in Hindi, it is called Hindi, and Urdu, it is also called alsi, and this plant has several uses, like the seeds are used for the seed oil, and also it has high, I'll tell you in the coming slides, high nutritional values, and the stem is used for the nylon fiber, and it is also has a industrial applications, several industrial applications like paint industry and all. Now you see linseed, you can see here there are two important components, nutritional components, which are very important and unique to linseed. That is the content of the omega-3, omega-6 and omega-3 fatty acid. Now, if you see the plant sources, among the plant sources, even the walnut is considered having high omega-3 fatty acid, but linseed is much higher than that. Similarly, the lignan, which are known to have the anti-cancer properties, are also very high in the flaxseed. You can see that the sesames are the known best source, but the linseed are having substantially high lignan content. So, because of these important properties, the linseed has gained a renowned interest now, in especially in the urban areas, and it is around the 14 billion dollar industry worldwide. India is one of the largest producer of the linseed, and it ranks sixth in the world, but the productivity is very less. It is much lower than the world average. And how in India the linseed is grown mainly in the rain-fed area, and where the water resources and the other resources are very less. So it is grown in the Rabi season, wherever the irrigation is not there. Where the irrigation is there, farmers take the wheat or the chickpea, but in the absence of that, they grow the linseed. So this is the crop of the resource poor farmers. So what are the important traits for the commercially profitable linseed cultivation for the farmers? Because in the it is grown in the Rabi season. So as the summer approaches, like in the February, the March, the temperature increases. So the yield quality, and like the oil quality and the yield also decreases. So the early maturating varieties are required. For that, early flowering, which facilitate early maturity, is important, and the high test seed weight and all these things are important for that. It is a long day, facultative long day plant like the Arabidopsis. So flowering in the early days is important, especially in the short days, because the long days start coming after December. Now, in the germplasm genomics, there are two approaches of biparental mapping, which can be used to identify what are the genes responsible for the quantitative trait, and other is the association mapping. So, focus on this lecture is the genome-wide association study. You know that it uses the population scale samples and the mapping population, and like in the biparental mapping, the recombinant only between the two parents, the extreme parents, are taken into consideration. But in the genome-wide association study, all the historic recombination which has occurred are taken into consideration. So the natural variation, natural diversity is the most important thing. And so that is why the chances of having the marker associated with the trait of variation is very high in the genome-wide association study. So this our work is part of a SERP project, early career research project, and some part of a DBT project. Now, as I earlier mentioned, that this is a rain-fed crop, and for although it is a facultative long day plant, but there is a genotypic variation there in the flowering. Okay, as you see, the two lines of germplasm lines of the linseed planted on the same day. One is at the flowering stage, and one is at the capsule formation stage. So there is a large variation for the trait of interest, these traits, and this is required for genome-wide association study. So what strategy we have used? I come from the Institute National Bureau of Plant Genetic Resources, and this institute conserves the germplasm reservation, germplasms of the crop variety of the cultivated crops. Okay. Now in the linseed, there are around 2,600 accessions which are conserved in that. That is throughout the world. And to shortlist that for this project, we have screened the earlier literature and whatever the evaluation was done earlier by the Eclipse and all that, and shortlisted around 220 accessions showing variation for the different traits like early flowering, early maturity, plant height, and the seed weight and all that. And from that evaluation over 5 equations, we shortlisted 131 accessions, and we genotyped them by genotyping by sequencing. And this evaluation was done at the 3 locations. Linseed is grown in India at the 3 agroecological regions, and we evaluated. Delhi is just near to 1 on 2. So we evaluated 3 times at the Delhi, or 3 continuous here, and 2 in Akola in the Maharashtra, which is in the zone 3 of linseed growing area. Now you can see here the variation. This is the violin plot. These are the environments, different environments where it is grown, and this is the variation. Now for the days to 5% flowering is the initiation of flowering. 50% is that the 50% plants flowered, and 95% that most of the plants of the accessions are flowered. Now you see the variation. The early flowering start as low as 35 days to around 90 days. That much variation for the initiation of flowering. Similarly, for the 50% and 95% flowering, there is quite large variation. For maturity also, you have a maturity at around 110 days to around 145 days. And this also differs based upon the environment. Like in the Delhi, the maturity is slightly late because it is a colder region, and in the central part of India, the maturity is little early. So that is for the plant height. You can say the plant height in Akola also it's slightly less, and the plant height starts something from 25 centimeter to as high 100 centimeter in all this location. So this phenotyping we have done in something called augmented block design, statistical design for the analysis. Okay. So now for the Jivas, you need 2 things. One, strong phenotyping, and then the genotyping. So we have used genotyping by sequencing approaches. We use the restriction enzyme, and then a subset of the genome is sequenced. And based upon that, we could get around 68,000 SNPs, high quality SNPs. And these are the distribution of the SNPs across 15 chromosomes. And these are the distribution of SNP across the individual chromosome. So this is important because we need to we do not know where the genes of our interests are located. So the distribution of the SNP should be throughout the chromosomes. So now this one important thing is that for the genome-wide association study is the genetic population genetic structure. So if the population structure is very distinct, there are the chances of having the false association. So it should not be much distinct. So we could, when we have studied the population structure using the SNPs, we could see there are the four population, but in each population there are intermixing. Like you can say that in this population one, the largest one, there is a blood of the population 2, population 3, and population 4. Similarly, for all 2, 3, and 4 population, there is a intermixing, and that is a suitable population for the association analysis. And this is a dendrogram of this 131 accessions. Now we have used the multilocus marker because ours is a quantitative trait, and there are certainly more than one gene or the marker associated are expected to be associated. So use multilocus markers, and this is called Manhattan plot. This plot indicates, like, these are colorful dots are the SNPs, and there is a threshold value here of LOD of 3, and the SNPs which are above this are considered significant SNPs. Since there are the chances of association, like false associations are also there. So we considered wherever the SNPs, either it should be identified. We have used 6 methods, of which the 5 methods identified the significant SNPs. So if the SNPs is to be considered genuine, it has to be repeated in more than 2 methods as well as more than 2 environments. So similarly, we here identified like 3% as the LOD for the significant SNPs, and to call the SNP stable, more than 2 methods or environment it should be there. So accordingly, for the flowering time, 3 flowering time trait, we could get 53 stable SNPs, 30 stable SNPs for the days to maturity, and the 27 stable SNPs, associated SNPs for plant height. And the SNPs which are in the pink dots, actually they are the SNPs identified by more than 2 methods. So these are the tabular information about the each associated SNPs. Now you see the yellow highlighted SNPs are the SNPs which have been identified by more than 2 environments and also more than 2 methods. So these are considered to be a strong and robust SNPs. Similarly, this is for the 50% flowering, and this is for the 95% flowering. This is for the days to maturity, and oh, sorry. Well, the important thing in this is that the SNPs, how much variation does it addressing the trait variation? Suppose we have talked that for the days to maturity, it is as low as like 100 days to 145 days. Okay. So what are the SNPs which could explain this variation? Okay, suppose there are biallelic SNPs. So which SNP is associated with the early phenotype, and which is in the late phenotype? So how much is explained? So in this, the what we have studied, we could get around 38% for the different traits, around 38% trait variation that was accounted. So These are the QTNs. QTNs are quantitative trait nucleotides. You must have heard about the quantitative trait loci, which is generally used in the biparental mapping population. And since this are used by the multilocus methods,
Projection is
is there. Okay, so these are for the plant type trait, the QTNs. Okay, it was the several in the different chromosomes. Now when we put them in on the chromosomes, these are like for the different traits like flowering trait it is in the blue color, maturity trait it is in the red, and the plant trait it is in the green color. So these are the details which are located on the chromosomes. Now, whatever the earlier studies have been done, whereby the biparental mapping or the GBAS, so we could get very close association. Like earlier identified mutants also were identified in this region. So two different mapping population fitting to the same gene or the marker is a kind of validation of the work what we do in identifying the gene marker. So this is a map, and the novel QTNs have also been identified, and the earlier QTNs have been reproduced here. So now this is linkage disequilibrium decay. It is a most important aspect in identifying the candidate gene. Suppose the marker is associated with certain trait. So how is the linkage decay? How is the recombination frequency, and how that is decaying? So that determines what is the region of the chromosome should be taken to be considered as a candidate gene. So when we did analysis, we found out that the 50% of decay is around the 30 kb region of the DC, and then that is why we took 30 kb up and the downstream of the significant SNPs, the stable SNPs, and then identified the candidate gene. Now these are the you see the important candidate genes. For the flowering, this is completely for the 5, 50, and 95% flowering. You see how interesting candidate genes, potential candidate genes are there. Like there are genes which are associated with the polar tube growth, for the polar nuclear fission in the pollination and the reproductive polar tube growth, regulation of the growth, microsporogenesis, mitotic cycle, pollen development, reproductive system. Ubiquitination is a crucial process which is involved in the flowering time, pollen germination, pollen development. So these are very potential candidate genes which have been identified for the flowering time trait in linseed. Similarly, for the days to maturity, there are important genes identified which have role in the seed growth, fatty acid biosynthesis. This is oil seed crop. So the role in that inside development, and like in the dormancy, female organ development, all that important genes have been identified. Similarly, for the plant height, also important genes have been identified which have known role like the chlorophyll, regulation of the growth, etc. So we hope that these genes are there to play important role. Now, having done this, the association study, we use the transcriptome analysis for that. So we use the early flowering accessions, two accessions, which are early flowering and maturing accessions, and we use the different tissues like the bud 1, bud 2, flower, leaf, and stem, because these tissues, vegetative tissues, are also important in perceiving and sending the signal, floral signal. So in this transcriptome study, around 30 transcriptomes were studied in this, and this is the total reads and the reads mapped on the genome, which are almost more than the 92% in all the cases, and these are the different gene expression in the different reproductive tissue against the vegetative tissue, either stem or the leaf tissue. Now this is a volcano plot showing what are the top. These are the labeled as the top differentially expressed genes either upregulated or downregulated. These are the representative comparisons we have taken for one maturing accessions. Now, this is called state law. This is what are the common genes which have been observed. Like we have taken two exceptions. So in the two exceptions, what are the common genes which are there upregulated so that we can confidently say these are involved in time regulation. We studied the cake pathway analysis where the genes have been already mapped. Uh, Known genes have been mapped, which have role in the flowering time regulation. And this is especially a circadian pathway, which is the part of the photoperiod pathway where the known regulators have been mapped in this and found differentially expressed. Similarly, in the hormone play important role in that, especially the ABA. And we had seen that the differential gene expression at the different levels. The redox process also we have found the significant gene expression. These are the enriched cake pathways which have been identified in this based upon the Arabidopsis work. This is the differential gene expression of the candidate gene which have been identified in the jivas. We try to validate that. And in the transcriptome, what are the most differentially expressed genes? That the top 100 genes we have shown here. So we can see both the accessions, the type of the gene differentially expressed are same. Means the similar pathway are playing role in the flowering time regulations. And these are the known genes which have been established in the Arabidopsis and what is their function, ortholog function in the gene seed. So that gene expression we have seen. Based upon Jiva studies, transcriptome analysis, and the known ortholog of the flowering, we identified two most potential candidates, and these are like flowering locus, like which is very known to origin, and one the AP2 ARN transcription factor, which is found to have role. We tried to study, we had done sequencing of early and the late accession, whole genome sequencing, and tried to see allelic variation, but these two genes did not show any allelic variation. So maybe the regulatory region or the other region might be having the variation, but the other key genes are show a quite good variation, like in this, like this AGL. You can see that the early accession is having one amino acid threonine and then the lysine. Similarly, in case of the Dilla protein, there is early trait specific variation in that. So we could find out the haplotype. So similarly, for the test seed weight, we had done the same thing. 1,000 seed weight important trait. The jivas evaluated at the 5 locations, identified the QTNs, significant QTNs, and then mapped them on the chromosomes, identified the region. Which are the QTN rich region of the chromosome that could be used for transferring this important trait. In this case, we could see the 1,000 seed trait variation starting from the 2.5 grams to 11 grams. And we also identified and registered the germplasm, which is having the highest seed weight in the linseed. So this is our important part, which as I discussed that how based upon the biallelic allele, if you what we have done here, we have divided our population based upon what allele it carries, and then we have taken the average weight of that. So we could see that if like in this case you see, so the accessions carrying some particular allele will have a different advantage, has the advantage over the accessions having the other allele, and that is significant in many cases. Okay. So this is desirable to transfer the important trait, and this shows that these alleles are genuine and can be used for the transfer of the trait in improving the quality. So, yes, I'm done, sir. Now this is the last slide. So this was for the 131 accession, but the National Gene Bank of India has around 2,600 accession. In our current project, we have genotyped whole genome by whole genome sequencing all the accessions, and that means we'll not discuss here, but we'll be doing that and studying the entire diversity by whole genome sequencing. So this is the summary. So this is our team. Not all team members are here, but 3 of my labs, 3 genius idiots are sitting here. Dania has done, he has done transcriptome studies. Ankit has done the Jiva study. Deepa has done the candidate gene, and Shraddha is sitting there. He has helped in the phenotyping and in the allele mining. So thank you so much. And,
One or two quick questions. We don't have much time left. Any quick question? If not, I will just ask one question. Like, have you got any correlation between the Jivas studies and your transcriptome study? Any gene which is the candidate which is having the role in the
yes, yes, two genes we identified.
two genes? So, what are you going to do further?
What am I going to do further? Now I will validate with the Cas marker, and then and also validate in the biobank, because there is a work going on the mapping population, and we'll validate on that, and then that will be used as a marker for the marker estate selection.
Any question? Quick question. One. Okay. Thank you, Prakash. Next. Again, mine.
Thank you, sir. Uh, as our next speaker, I welcome Dr. Prashant Anupama Mohan Pawar to the stage. Dr. Prashant is an associate professor at Regional Center for Biotechnology, RCB, Faridabad. He is a recipient of DST Inspire Faculty Award and has been recognized with the prestigious Energy Bioscience Overseas Fellowship 2019 by the Department of Biotechnology, Government of India. Dr. Pawar specializes in cell wall biosynthesis, cell wall remodeling, tailoring cell walls, and oligosaccharide signaling. Dr. Pawar has an extensive publication record with over 30 research papers in high-impact journals and holds a patent reflecting his significant contributions to plant biotechnology and cell wall biology. His topic will be building a wall for sustainable conversion lignocellulosic biomass to value-added products through in plants through cell wall engineering. Sir, the stage is over to you.
Thank you, ma'am, for kind introduction, and I would also like to thank organizer for giving me this opportunity to present my work. So, my lab actually works on plant cell wall, and today I'm going to talk about the strategies to engineer the plant cell wall so that we can improve the lignocellulosic biomass properties. Okay, so we'll straightly go to what is lignocellulosic biomass. So lignocellulosic biomass can be from the agricultural residue, something like a rice straw. Okay, it can be from forest residues as well, right? Which is lying on the in the forest. At the same time, forest trees such as bamboo and poplar can be grown in a controlled manner so that they can be utilized for bioenergy production. So what is actually the advantage of using this lignocellulosic biomass? First of all, it is renewable source. Plants can be grown again and again. Second, Dr. Danta also talked about that it is free from food to fuel debate, right? Like for generation fuel, we are not using the seeds. Okay. So the third is like it is a kind of carbon neutral energy in the sense that the fossil fuel adds extra carbon to the environment, right? But trees generally captures it and it can recycles it. So we can say it's a carbon neutral approach, right? And it is abundantly available. It doesn't need extra cost to grow, right? And it has variety of application. And today I'm specifically going to talk about the bioethanol production application, right? The linseed Doctor talked about also, it has a good fiber quality as well, so it can be used for making biomaterials and so on, right? So it has variety of applications. Okay, so let's talk about the lignocellulosic biomass. What is exactly that? The lignocellulose word comes from lignin and cellulose, right? And that is a fundamental component of the cell wall. So this is actually a cross section from the Arabidopsis stem. As you can see, it is stained with toluidine blue. It stains specifically cell wall. Okay, so in the blue these are xylem cells, and in the pink these are parenchymatous cells. So as you can see, the xylem cells are more thicker and the parenchymatous cells are more thinner, right? So the message I want to give here is that the cell type has a different cell wall composition. But just for the sake of this talk, we can consider that the cell wall in generally composed of cellulose hemicellulose. Remember, one of the major hemicellulose is xylan, pectin, and lignin. Okay, so in order to engineer the cell wall for a variety of application, what are the major challenges? Right? Let's talk about that. So we actually don't know how these cell wall components are synthesized. Like what is the molecular mechanism? What are the genes which are involved in the regulation of cell wall and so on. Second, the synthesis is another part, but they also interact with each other, right? So we don't know how they interact with each other once they are deposited extracellularly, right? So the interaction is unknown. And third, when plant gets attacked by pathogen or by by giving kinds of stress, how they modulate themselves, or when we modify the cell wall, how they remodel, what are the cues and all that we don't know, right? So in my lab, to address these challenges, we worked on three different aspects of cell wall biology. We try to understand what are the genes or transcription factors which are responsible for cell wall biosynthesis. uh, second part is we would like to fine tune the cell wall structure so that we can improve the cell wall properties or lignocellulosic biomass properties. And third is we use omics based approach to identify the genes which are responsible for cell wall biosynthesis, and then we functionally validate them, and then we subsequently use those genes again for the manipulation. For this talk, I'm going to talk about this part here we where we would like to re-engineer the cell wall, right? To improve its properties, right? So before going into what type of modification, I want to talk about the process of bioethanol production. So generally, we have the biomass. Cell wall, as I said, it's a complex matrix. So we need to loosen the cell wall, right? So we use some chemical treatment like alkali acids to loosen the cell wall so that enzymes can go inside or sit on the polysaccharides, right? So the process of degrading the cell wall using enzymes, we call that as a saccharification. And the sugars which are released are used by Saccharomyces as the energy, and they produce ethanol, right? So this is the process, but this is not cost effective process because we need expensive enzyme, or we also use harsh chemical to degrade the cell wall, which is not good for the reactor, right? So two major hurdles are there. There are others, but I'm going to talk about lignin and xylan. So lignin is basically a phenolic polymer, which is a high molecular weight polymer. It is difficult to degrade, right? Even 72% H2SO4, if you use, it's difficult to retain with that high acidic, H2SO4. At the same time, xylan, which is a shorter polysaccharide, but interacts with other polysaccharide and makes cell wall more stronger. So I want you to remember two things: the lignin and the xylan, and the acetylation which is present on the xylan. So these acetyl groups are also hindrance for the cell wall digestion. Okay, so let's talk about the lignin and xylan in short. So lignin is synthesized via phenylpropionate pathway, and the amino acid phenylalanine is the precursor in Arabidopsis. In monocots, phenylalanine and tyrosine are the precursor, and from that the monolignols are synthesized. This whole process happens in the cytoplasm, and then these monolignols are transported to the cell wall where they are polymerized. Okay, and just remember that the plant which I'm talking about, the composition of the lignin is different, right? So this H lignin, G lignin, and S lignin has different composition. Okay. The another polysaccharide which I am going to talk about is xylan, right? So xylan is a shorter polysaccharide, something around 100 to 200 degree of polymerization in the Arabidopsis, and it is heavily acetylated. Okay, so the simple hypothesis was to alter the xylan as well as lignin structure for improving, for example, bioethanol production, right? So the strategy was to mutate this particular gene F5H. So if you block F5H, the flux will go towards the G lignin, right? So we had one mutant, Arabidopsis mutant, where there was a mutation which made the FAH plant non-functional, right? So the plant produced G lignin. The another strategy was to express the xylan acetyl esterase, right? So this was the enzyme which deacidify the acetyl group, and then it will reduce the acetyl content, right? So the strategy was simple. So then these are actual plants. So this is Arabidopsis white plant. This is a plant where there is a mutation in FAH1 gene. This is the plant where we have overexpressed acetyl xylan esterase, and this is a cross between these two. So one of the advantage of using Arabidopsis is we can cross the plant and make the hybrid plant very easily, right? So this is a cross between these two, right? So from now on, I will call these two as a parent plant. This is the wild type, and this is the cross plant. Okay. So now when you overexpress the acetyl esterase, when you express the esterase, the acetyl content should be less, right? So then we looked at that whether the acetyl content is less. So as expected, the esterase expressing plant has less acetyl content, and also cross plant has less acetyl content. But FAH1 and wild type, naturally they did not had less acetyl content as compared to the AXE expressing plant. Okay. The second confirmation we wanted to do for the FAH1. Okay. So as I told, FAH1 plant will mostly produce G lignin, and fortunately we have a dye which can sense the G lignin. Okay. So so this is actually the plant where we made the cross section and we stained it with the mole. Okay. So so mole basically stands S lignin. Okay. And in the wild type plant, in the fiber cells, there is more S lignin. So as you can see, the wild type plant as well as the AXE plant, which has more S lignin, those are, take my word for it, the pictures are not clear, those are orangish in color. Okay. And the FAH1 plant and the cross plant had yellow color. Okay. This suggests that these plants, the overall message from this slide is that we phenotypically confirm that whatever modifications we have done are correct. Okay. Now we wanted to see the effect of such modification on other cell wall component, right? I told you that there are celluloses, lignin, the monosugars which are coming from the hemicelluloses. So except the total sugar and cellulose content, the monosugar content was not affected. As you can see, just remember the xylose content was not affected, which is coming from the xylene. So the cellulose content was increased only in the cross plant, not in the parent or in the wild type. Okay, so if you remove the acetylation, I told you that it improves the digestibility of the cell wall. So this is the paper where they found that when you use the acetylxylanesterase and xylanase break the backbone, the oligosaccharides are more and the xylose release is more. Okay, so we wanted to test that. So what we did actually, we took the cell wall from the plant or the powder from the plant. We digested naturally that with the xylanase. The xylo-oligosaccharides were purified because it had buffer component and other components as well. And then we did the mass spec analysis, right? So I'm really sorry for the units; these are very small, but basically this is the intensity on the y-axis, and this is the m by z, and these peaks basically are the xylo-oligosaccharide. Xylo-oligosaccharide. Okay, so just the red arrow, just concentrate on that. This Okay. So this red arrow represents the xylobios. So as you can see, the xylobios peak is very less in the wild type, but more in FAH1 plants, also in the HC expressing line and the cross plant. This suggests that the xylan digestibility is increased, right? So we quantified this, and as you can see, the xylobios peaks are more, and also the shorter chain oligosaccharides are more and the longer chain are less. This further validated that the digestibility is increased in cross plant as well as in parent plant as compared to the wild type plant. Okay, so another thing is that when you digest it with the xylanase, you can also measure the xylose. Okay, that is also the measuring xylose is another strategy to see whether the digestibility is increased or not. So as you can see, without treatment and with treatment with glucuronidase esterase, we found increase in xylose release in the parent as well as cross line, right? Not only that, this is a enzymatic digestion, right? So we extracted the xylan using alkali, okay, which extracts the hemicellulosic fraction, and we probed it using the antibodies which are specific for the xylan, and the chemical extraction was also easier in the cross as well as in the parent plant. Okay, so the xylan digestibility as well as extractability was increased in the cell wall modified line. Now, as you know, the cellulose is also main component of the lignocellulosic biomass. We tested its digestibility. and we found without treatment there is increase in glucose release. This is because there is increase in cellulose content. Okay, but with treatment as well, we found increase in the cellulose digestibility. This suggests that there are some modification in the cell wall which we don't know that has triggered the digestion of more digestion of the cellulose. Okay, and on the top of that, when you did all these digestion and when we measured the pellet had less lignin in cell wall modified line as compared to the wild type. This suggests the lignin solubility, which is a high molecular weight polymer, was also increased in these lines. Okay. So this is my last result slide. So if you remember that in the cross plant we had increase in cellulose content, right? So if there are some changes in the cell wall, then those are pursued at the plasma membrane, and that basically triggers the signaling, and that might increase the expression. So we tested the expression of these genes. So these CESAs are cellulose synthase genes, which are responsible for cellulose biosynthesis. Those were increased only in the cross plant, and the Thesis one and wax, these are sensors on the cell wall which senses these signals in the cell wall, and they can transduce the signal, and those were also increased, right? So this is kind of a possible explanation for increase in the cellulose content, but we still don't know. Okay, so the take home message from this my presentation is it is possible to certainly modify the plant cell wall without any effect on the plant growth. Simultaneous modification of both lignin and xylan did not increase the saccharification efficiency, but individual modifications were sufficient to improve the lignocellulosic biomass property. And we also found that there is activation of cell wall integrity signaling, and which needs to be further investigated. Importantly, this is what is uh, this is what is the model which we for the xylan digestibility which we have built. So these are wild type plant. The black ones are basically the acetyl group, and the these red symbols are basically xylanase enzyme. So as compared to the wild type, the xylan digestibility is increased in the 35S ANXU esterase expressing line because there was less acetylation. And for the G lignin, it was actually surprising the digestibility was increased. But we think that the proximity between the xylan and lignin was less. That's why in this G lignin rich plant, the xylan digestibility was more, but we need to further investigate. So this was published last year in Biotechnology for Biofuels. So this is my team which do all the hard work so that I can speak here. So Aniket did most of the work, and Anand is in the audience who also help Aniket, and Ravi also help. And these are other people who are working on other aspects of cell wall biology about synthesis, metabolism, and so on. And this I would like to thank funding agencies as well for generously funding.
Thank you, sir, for such an interesting talk. I open it for any discussion, any questions? Any questions from the audience? Sir, I have a question, please. So when we are modifying the lignin degradability properties and all, when we actually transfer it to the plant, will it result in any kind of like mechanical strength or effect on the mechanical strength of the plant, like when it is growing?
Yeah.
At that time, will there be any
So
effect?
in this, we haven't tested the mechanical strength, but the morphology of the xylem cells was not affected. The plants were growing fine. So I think that it was not, affected. So more importantly, we here did not modify the lignin content, but the composition, right? So if you modify the composition, then it is tolerated by plant. But if you modify reduce the lignin content, it is not tolerated.
Then it can be an issue.
Right, because for example, we have the plant with G lignin, and the conifers have high G lignin. So plant has natural
So we're just replacing one
replacing
lignin
one
with
lignin
another.
another, right? So it doesn't actually affect the strength.
Thanks. Any other?
Does it have any effect on, like you haven't checked, but any effect on the disease?
Yeah, very good question. Yeah. So we tested against the Pseudomonas. So there was no change, at least in the cross plant as well as in the parent plant. In fact, we are working on other aspect of it. We think that in some of the mutant we see enhanced plant immunity, because there is activation of a basal immune response from the plant. Something like a acetylxylanesterase expressing plant, there is activation of oligosaccharide signaling, and then the basal immunity markers were upregulated. So at least for these modified lines, we did not see any effect. Yeah, but it could be different for different kind of pathogens. At least for Pseudomonas, it would be. Yes. So G-lignin is basically coming from the coniferyl alcohol derivative, okay, and so the G-lignin is coming from coniferyl alcohol. Coniferyl alcohol is synthesized in cytoplasm, and when it went to the cell wall where it is polymerized, and then it is called as a G-lignin. So after polymerization, we call that as a G-lignin.
Thank you so much, sir. I would now like to welcome our next speaker, Professor Praveen Verma. Sir has already been introduced to all of us as he is also the chairperson for this session. His talk will be on mechanism of fungi, overcoming cell wall defenses to cause disease in plants. Sir, the dais is over to you.
Thank you. Thank you, Dr. Kakuli. So, I, क्या हुआ? Where is it? So, today's topic, what I have taken is one part of my work. This is a small part of my work which has recently got published. So, but that will be really interesting to all of you to understand the basis of plant and pathogen interaction. This one. Should I use my own laptop? Let me go. I'm sorry, my throat is very bad, and I may take a little bit more time because of my bad throat. Yeah. Good. So the topic I have taken today is, that my previous speaker he has talked about cell wall and lignin. So I will, my talk will try to understand how fungal, pathogens they actually, they actually solubilize or they will stop sometimes, the lignin biosynthesis pathway. So that I will, thanks to him that he has already discussed about the pathway which leads to the lignin biosynthesis. So usually in the plant and pathogen interaction, when we talk about the, if you all are from the plant science, usually we teach plant pathology or the host cells. But usually, in most of the cases, this is a fine tuning between the plant and pathogen, where the plant immune response is always trying to counter the intruding pathogens, and pathogen use weapons to suppress such responses. So, what are these weapons? We will discuss. So, if plant wins, there will be no disease. If pathogen wins, there will be disease. So, this is the major things which is coming out right now. So, this is the new concept which is coming is the effectors. So they are actually the secreted effectors are the pathogen weapons. Just like a weapons, they actually try to impose in or introduce inside the cell to stop the defense pathway. And this, from my today's presentation, you will be able to understand that how it is actually doing all these things. So there is a this is it's a reason behind the virulence and host jump. You all know that the different degree of virulence of different pathogens are there, and this is also like you must have heard that the variety was earlier resistant to the certain pathogen, but now it has become susceptible. So these type of things happening because of these effectors. So there are two types of effector we call. One is the apoplastic effector, which actually secreted, and the name itself suggests that it is in the extracellular space. It is secreted out from the fungus. And second thing is that cytoplasmic effector, that it is secreted outside of the fungus and then it is entering into the host cell. So here, they are crossing one membrane, that is the fungal cell membrane. Here they are crossing two membranes. So this is complete mechanism is there. So that you will try to understand. So the lifestyle of the pathogen is of two types. One we call is a biotrophic pathogen. Another we call as a necrotrophic pathogen. Biotrophic pathogens, they actually live in harmony and they extract their nutrition from the living tissue. And necrotrophic, they are actually the dead. They, they take the nutrition from the dead tissue. So they promote cell death. So usually it is known that the biotroph they retain harmony and necrotroph has smash and grab. The work that I am going to present is the first work of this kind where necrotroph has been also shown that it is also having a some sort of the the mechanism of the infection like biotroph, and this is that we have shown in this particular thing. So the paper we have published recently, we have got one the commentary of this particular paper. So the topic is very interesting, that return of chickpea zombies. So they have talking about, because it takes the nutrition from the dead tissue. So they have a return for chickpea zombies. How Scopeta really overcomes the cell wall defenses and qualities. So this I'm going to discuss about these things and today's work, the Scopeta story, an example of discovering virulence mechanism in the necrotroph. So this particular disease is infecting the chickpea Sisal erythrinaeum, and it is caused by Scopeta. It's a necrotrophic fungal pathogen, and it is most of the cultivated varieties and more are mostly susceptible. Although we have now doing breeding for this some of the variety that we have got, and we have published some of the things. But the important thing is that it actually affects all the aerial parts. Recently, we have published this as a pathogen profile in molecular plant pathogen, where you can just see that these are some of the suitable time. It actually destroy the entire crop, and mostly the 100% loss in most of the times. So we have started our work with two things. One, we started with de novo genome sequencing long back in 2012 that we have started this work, and we have done this genome sequencing of this particular pathogen. And later on, what we have done is that we have also done the transcriptome sequencing of this particular pathogen during the infection. And this recently we have published in NPMI, and where we have got many of these type of proteins which are involved in the resistance that we have got in our case. So the story started with this particular protein long back. This is for a student that I am telling you that when we have got one of the protein which is coming from which we have identified in the transcriptome. That time we used to do the not the modern type of transcriptome, but the SSH. This is called suppressor subtractive habitation. So what has happened, like we have got one gene which was hypothetical. It was not having any relation, anything from anywhere. So we thought that but the expression was very high during the infection. So from that reason, how we have proceeded, you'll see in this particular story. So after the sequencing, we have found many of the motifs which is already known in this particular thing. So, pathogen, we have got one of the protein we have named as a PIC25, and this is having one motif which is RX, LX, D, E, B, or Q. That means it is called, why it is called the it's the RLXR motif, because this is having a similar sequence which is known in the plasmodium. Plasmodium also do have whenever it sticks in the blood cells, it actually forms a parasitophorous vacuolar membrane, and this motif is also present in the parasitophorous vacuolar membrane. So we have main worked for many things. We have also proven that it is working in the plasmodium also. This same thing is working in plasmodium also with the help of one of my friend in National Institute of Immunology. And this the for this region, we have started this work with this. You can just see that after this 0 hour and this 12 hour of infection, the gene expression goes near about 500 times. So that means it must be having some role in this. And Dr. Kunal is right now principal scientist at IHBT Palampur. He has worked and got this claim. So this the PEC25 that we have given it, that is this is basically Excel life effect candidate 25. This is just like a name that we have given. So this is a novel effector protein. So Srinivas, Ankita, Kunal, and Sandhya all four worked in this particular thing. As Dhampakas is knowing most of them. And the important thing is it is having one Excel motif in the 5' region. There is a single peptide one also there. And then we have got some of the reason which is arginine rich motif. So what arginine rich motif is doing that we have started doing. So the experimental proof that it is an effector protein. So for that we have gone for the effector secretion. You can just see that if it is in the external environment, it doesn't get secreted. You all know that the protein synthesis is an energy consuming process. So it will not be produced that particular time, and it is not getting secreted. So it will be secreted only when the plant is there. And then what we have done is that we have done in the transgenic culture, and from there we have done the SDS analysis, and we have found that Western blot analysis, we have found that this particular protein is indeed it is secreting. Where you can see that it is secreting out from the fungus. That was of our first goal. It was there. We have also proven by several other methods, like we have gone for the LCM SMS analysis, and we have also identified that this is a secreted protein out of from there. Also, we have proven with it's called yeast secretion trap assay. So there we have shown that this particular protein, when in the yeast also, it is getting secreted out. These are some of the different methods that is important. So now the thing is that it is secreted. So where it is going, whether it's going to a host cell or not. For that, what we have done is that we have expressed one protein at 25 with Y80. There is yeast yellow fluorescent protein, and then we have used this transformed fungus to infect the plant. And what we have found that it is is what we have got is that mostly the things is that it is going to the host and localizing into the nucleus of the host. That was really exciting for all of us. And you can see that it is actually localizing into the nucleus of the host plant. So that's why we got excited. That means you can just imagine that one fungal protein, it is coming out from the fungus and going to the plant cell and localizing into its nucleus. So let's see what is going on in this regard. So what we have done is that we have also done the knockout analysis, and knockout we have found that the white type, the infection is very high, but if we do the knockout, that infection goes down. And also when we have used that NES, that is nuclear localization signal, nuclear export signal, sorry, nuclear export signal. In that case also, that the infection goes down. What does it mean? It means that the localization to the host nucleus is very important for the fungal infection. So now, up to now, you have understood that in the external environment, it is coming out. Now it is the new thing that it is coming out from the fungus and going to the nucleus of the host plant. So this is quite interesting. So, but what we have got, and then our next question was, if it is localized into the host nucleus, so then it must be interacting and manipulating a transcription factor or the transcription regulation. So for this purpose, what we have done is that we have gone for it's called tap tag analysis. Uh, Tap tag analysis that we have gone. So it's a tandem affinity prefixure. So what we have done is that in the column we have got this protein bound, and then we have total lysate that we have of plant lysate we have taken for that. And surprisingly, we have got one of the transcription factor which is actually interacting with this particular protein, and that was the CA beta lim 1A protein. It is known, many for many things that it is known that we have done when we pull down and interaction is to have read studies. All these studies that we have done. I'm not going into detail of that, but importantly, that these interactions we have proven by in plant also by this called by molecular fluorescence complementation, and also by plate analysis that we have proven by these two have been. So all 4 5 different type of things that we have proven that this particular protein is localized into the nucleus and interacting with this particular transcription factor. So this transcription factor is actually one of the genes that Prashant has already talked about. Is the PAL is one first enzyme of the phenylpropionate biosynthetic pathway. Usually, the first enzyme of the any pathway is rate limiting. So the what we have found that there the CA beta link protein, the transcription factor actually binds to the promoter sequence of this PAL gene. And that we have shown by different methods that it's and what we have right now we have got is that is basically when we got the C beta link, what we have got is that this is basically regulating the C beta link, and the important thing is that how this particular the agro infiltration that we have done with different aspects that we have done. I'm not going into the detail of that, but there is a there one essay we do have is called as a luciferase based essay. Very good and interesting result in this case that when this interaction with the promoter sequence of PAL promoter is actually having the problem when it is having the PEC25 also in the system. So that means somehow this PEC25 is actually preventing this transcription factor to bind to the promoter of PAL, the phenylalanine. So this is a complete story that we have got in this regard, and this we have got for many the disciplines say that analysis that we have found all these data. We have also done the different the fractionation of this protein and then the gel direction. And what we have found that in the case of when it is PEC25 is also there with is there, the binding of the protein to the promoter sequence is getting depleted. So that means that it is actually hampering in those things. So now the question is that it is hampering the phenylpropionate biosynthetic pathway. So important thing is that phenylpropionate biosynthetic pathway leads to the synthesis of many things that Prashant has told about the lignin biosynthesis, some of the phenols, polyphenols, the flavonoids, isoflavonoids, all those proteins are there is actually getting synthesized by those this particular pathway. So which is actually important for all of us. So for that we have gone for the metabolome analysis, one of the Ashutosh Pandey. And when we have analyzed all these things, we have analyzed all the different parameters. What we have found that it is actually lignin pathway that it is actually hampering. So the ultimate thing is the lignin pathway, which is getting suppressed by this particular effector protein. So this is the complete story that we have got, and you can just see that in normal condition, this transition factor it remains into the cytoplasm, move into the nucleus, bind to the promoter, and then secondary metabolite synthesis will be there, and there will be no infection or less infection. In the case of, you can just see that when we will have the Scorecata infection, so in that case, you can just see that it is coming, you can just see ये इधर हट गया, ये इधर देख लेने दो, तो 1 minute first. So now you can just see that these effector protein in the pathogen, and this will come out and go to the host cell. It is in the cytoplasmic region, and then it will be interacting with this linked domain protein, and this is how it is actually having the problem in the all biosynthetic pathway, and then this will be promoting the disease. So this is the one way that host cell is designing. So this paper has been recently published in Plant Cell, and this is made in India one. Nothing has been done outside. So the DPL effector K25 from necrotrophic fungal histopathology targets the kidney transcription factor beta one and negatively modulates the lignin activity, increasing the persistence. So this we have got recently, and this is our GNU. It's a beautiful campus from the top, and thank you very much. I also thank all my teachers and family members.
Thank you, sir, for this interesting talk. Questions, please.
Yeah, please. Plant inside plant. There are one paper has been published in Cell long back in 2010 by Kale. So that there they have shown that it is having the they take actually lipid wrap of the cell membrane, and it enters. They have also found out that PIP3 is involved in the transport of that. It is not, that there's no hole as such. So they actually goes like that. So now we have done also the NMR analysis, and what we have found that this protein is basically say it's an unstructured type of protein, and it is having a hinge region, and it is having a region just like horse, and so it is probably it is also taking the same way, but that paper has got controversy. Now, what we are thinking that our N-terminal, C-terminal region having some protein which we have mutated, and we have found that then it is not getting translocated. They are positively charged molecules. So they may be involved in the transport, but still it is not known. For any pathogen, forget about my pathogen. For any pathogen, it is not known. No, but penetration tag is also there here. But here is also there. But their effector delivery and crossing the cell membrane is still not. We thought initially that pexel motif, which is present in the plasmodium, may be involved in that. We have found that same like pexel motif is working in the plasmodium, but from our gene, but it is not working in the when we have mutated, it is not giving the same result in that. So maybe they have both of them have evolved separately, right? But somehow it has got some meaning in all those things. See, whenever there is a attack of any pathogen, so plant try to make the focus. So lignin biosynthesis and this, initially you must have seen that this pathogen is it's a necrotroph, so it goes through the hypoplastic space, like you move intracellular space. So intracellular space is the mostly hot spot of deposition of the lignin. So lignin, cannose, all those things are getting deported. So that's why maybe it is preventing that. So that's why we are getting a very drastic result when we get the knockout of this. Yeah. We have measured each and every the HFA, lignin, S-lignin, all those I have found. And we have found that few of the components, like synergic acid, synergic acid, those and another is ferulic acid. I'm not expert on that. So that is getting down.
So is it at the metabolite level that
maybe
antimicrobial?
it's just basically what we feel is that it is actually preventing the pathogen to spread.
for some of the upstream metabolites like cinnamic acid.
Correct. It is. We have checked for that too.
They have also antimicrobial.
Yeah.
And what I'm trying to understand is because of the lignin level or the metabolite level.
Actually, lignin, after the lignin sequence, usually, necrotrophic primary pathogen, they don't make the hostodia. They do have the penetration rate, but they don't make the hostodia. So they do have a very small episodia also. So they actually try to move between the intracellular spaces. So there the lignin deposition is more. So lignin deposition is less, then it will be easier for them to spread. So that is our theory that we have done. So any other? Yeah.
Wonderful, doctor. I've seen the progression of this work.
Yeah.
So I was just wondering, like the target transcription factor. So, did you check of the resistant varieties?
Yeah, it's a very good question. So when we have already done the QTL-seq analysis, and we have published in Plant Cell Environment. You must have seen that paper, and also across genetics that we have published. So there, what we have found that we got one of the gene which is present in QTL1, and that is basically say 80 hook like transcription factor. Now what we are working, we have got that this effector protein is also hampering that QTL protein, that candidate protein that we have done. So that is also a transcription factor.
Thank you, sir.
Thank you.
We move forward. I now welcome Professor Amit Kharkwal. Sir is the Deputy Director and Professor at the Amity Institute of Microbial Technology, Amity University, Noida. With extensive experience in microbial and plant sciences, he has led multiple research projects funded by prestigious agencies like DRDO, ICAR, NMPB, and DBT. His research expertise lies in plant microbial interactions, microbe interactions, and conservation biology. His work has contributed significantly to understanding symbiotic relationships in ecological and agricultural systems. He is a member of the Association of Microbiologists of India and has authored over 50 research publications in reputed journals, along with a book and 3 patents, underscoring his contributions to microbiology and biotechnology. So, his topic for today's talk is Talaromyces alba B verticillus, a novel plant growth promoting root endophyte with industrial and agricultural applications. Sir, the dais is yours. Thank you.
How much time I have?
Yeah, 20 minutes.
Right. So, good evening and welcome all of you. It's my pleasure to be invited by Professor MJ Abdi, my teacher. And today I am going to speak about a journey of our discovery from ecological niche of India. And I'll show you that how we can contribute towards regenerative agriculture and sustainable agriculture. So I speak about a species Talaromyces alba verticillus HNB9 strain. This species is very well known from the deep sea to the AC ducts as a saprophyte on fruits. So, but this strain which we have isolated from the Kandi region of Punjab is special because it is isolated from inside the plant root, and this has exhibited various multiple plant growth promoting activities, and I'm going to speak about its industrial and agricultural applications. So this species is a patented, agenically cultivated novel root colonizing fungal strain, and this colonization of plant roots by this fungus results in growth enhancement and resistance against stress, including biotic and abiotic stress. And its PGP activities include, which we have documented, phosphate, zinc, and silica solubilization and auxin production. And this also produces a beautiful red color compound, which you will see in the next slide. So this how this is we have documented in various medium, and This is Teloromyces, nothing but a sexual form of Penicillium species. And but this we have isolated and we have filed the patent and we have transferred the technology to one Delhi-based startup and another technology on its consortia, which I will discuss later. And this fungus, is having multiple plant growth promoting properties, as you can see from here. And up close, you see zinc solubilization, phosphate solubilization, silica solubilization, and IA production. And one of the concern in regenerative agriculture is that we are using a lot of fungi, but many of these fungi are mycotoxin producing also. So we did a test on this, and this is non-mycotoxin producing fungi, so safe for human and agricultural use as such. So we did a couple of tests like microscopy study, confocal imaging, and described the structure in detail, confocal microscopy, SEM studies. So we have described this all these things in great detail in publication. So you can have a look at this. And we did its molecular characterization using multi-gene phylogeny involving 4 genes, partial sequences, ITS, PEN-A, calmodulin, and RPB2 genes. And based on the sequence alignment, we were able to place this fungus as Teloromyces alboviverticilius. You can see Teloromyces rubilficans also, but these are two synonym fungus as per the literature. So I prefer the name alboviverticillus uh, than the rubilficans for some different aspects. So this you can say because it is forming the spores which are ellipsoidal in nature. So alboviverticillus is more appropriate name. So we come to the plant growth promotion stage. So this fungus basically, if you see, we did the seedling toxicity test. The seedling, it was not toxic to the seedling, and then we did the greenhouse test. As you can see, only the interaction, other things are common. It is pronouncing the root growth in the greenhouse, and then we come to the application to the field also. As you can see, we have developed the bioformulation also, wettable bioformulation, having the spore count of 5 into 10 to the power 7 to 9 per gram of the formulation, and this can be directly applied to seeds, seedlings, and standing crops. And we have done a lot of field trials all over the India, and this is up close how it looks like. So this was our first field application in Yamuna Father in Delhi, and this fungus not only promotes plant growth but early flowering and early fruiting. And this is how it colonizes the roots. The spores are colonizing the roots, and that is how it gives the plant promoting activities. It promotes the absorption of minerals. I will show you. Now, this experiment was conducted in Punjab Agriculture University and on 3 different varieties of wheat. And what we found that overall, there was 13.3% enhancement in yield in wheat as compared to control across 3 different varieties, Unnat, PW343, and XT3086. And similarly, we did a trial in Bulandshahr in Pusa Basmati 1121 variety, and we got 33% yield enhancement. This was feedback given to us by the farmer. And now what we have done, we have developed the consortium of this fungus with other plant growth promoting rhizobacteria. So this is Bacillus subtilis, as you can see in the dual culture, it grows along with the fungus, and we tested this consortium in the greenhouse, and we got significant enhancement in leaf, root length, fresh weight, dry weight, and total chlorophyll content. And here you can see the picture also. And the root colonization with the Bacillus subtilis of the fungus increased significantly. Up close, you can see the fungal spores colonizing the roots. And then what we did, we co-cultivated the fungus and bacterium together. And to my surprise, the bacterium was causing the sporulation by swelling the hyphae of the fungus. Here you can see up close. And we analyzed the spores. The spore size was significantly more in Saccharomyces plus Bacillus combination. And also the spore production was more in Saccharomyces and Bacillus combination, culture. And then what we did, we applied this consortia in field using HSP9 alone and HSP9 plus Bacillus with the defined spore count. And this is how we treated the rice plant. And this is a field near Faridabad. and you can see the yield of 18.34 quintal, 26.26 quintal in HNV9, and with bacillus it further increased. And similarly, this consortia we applied under restricted irrigation condition both in lab and field, and what we got results in 100% irrigation, as you can see. But what is more exciting is 50% irrigation, right? 50% irrigation, the plants have similar parameters as compared to 100% irrigation. And even in the restricted irrigation, the plants were able to grow. And we did the ICPMS and PK analysis of these plants and found that the treated plants with fungus and fungus plus bacteria had more NPK content as compared to control plant across all the conditions in both shoot and root. And this work was conducted in field again in PAU under three conditions: restricted irrigation, full irrigation, and rain-fed irrigation. So full irrigation means 6 cycles of prescribed irrigation. Restricted irrigation means 3 cycles of irrigation. So 1 cycle of irrigation is skipped in between. And rain-fed is no irrigation, only rainwater irrigation that is available from the Mother Nature. Now, if you see in this also, the yield in the restricted irrigation in the HN9 alone and with the Bacillus subtilis is more than uninoculated, but in the full irrigation also, if you see, it is restricted irrigation is more than uninoculated full irrigation. So we can go for reducing this cycle of irrigation directly, and 50% irrigation will give the result of 100% irrigation. Similarly, the effective tillers, the full irrigation, 338 without treatment, okay, and restricted irrigation 362 and 369, right? So again, this showed that even restricted irrigation is, useful with the application of this fungus. And again, you can see 1,000 grain weight. What was surprising, although the yield in the restricted irrigation was less, but the 1,000 grain weight, you can see with the fungus and fungus bacteria, was comparable to the 1,000 grain weight of full irrigation, right? So there was no compromise in the quality of the seed. Up close, we tested this combination for its silica solubilization activity, and what we found that the combination of fungus plus bacteria was enhancing the silica solubilizing activity, and all of us know that the available silicon in the cell wall has its role in the resistance against various types of biotic stresses. Now, this is not the only bacterium. Another bacterium isolated by one of my students, Enterobacter cloacae, CS1 strain, SK4 strain, from coal mines of Sikkim. So she is basically working in the area of heavy metal tolerance. So what we thought that let us combine this bacterium with the fungus. The HNB9 is strong phosphate solubilizer, and Enterobacter cloacae is strong ammonia production and also having activity for potassium solubilization. So we did the dual culture, and we are standardizing the co-culture right now. And here you see, HNB9 is not solubilizing the potassium in the Alexandrov medium, but this bacterium plus fungus is enhanced, doing the enhanced solubilization of potassium in the Alexandrov medium, and also oxygen production and ammonia production. So this combination we prepared for a bio NPK fertilizer, and we are developing this, and these are the early greenhouse results. As you can see, combination is increasing the growth and chlorophyll, and we have done the again NPK analysis, and this is increasing the total NPK content in the plant. And similarly, we did in another plant, that is fenugreek. Again, we got exciting results, plant growth promotion and chlorophyll content. And again, you can see the NPK analysis, fenugreek, right? It is increasing in the plant sample and also in the soil sample. And because this bacterium is from the mine area, is from Sikkim, right? So with Otherwise, it is tolerant up to 3000 ppm of arsenic, the bacterium. But with HNB9 also, up to 300 ppm, the fungus and bacterium can grow together. Another consortium which we made is protozoa plus HNB consortium. Soil protozoa are known to affect the microbial population in soil, and what they do, they exert selection pressure. And when soil protozoa are present in the soil, this selection pressure results in lysis of the PGPR, but also stimulate their growth. The lysed material comes to the soil and helps in the total enzymatic activity of the soil. So what we have done, we have tried to make a consortia with three protozoan species, HNB9 plus Bacillus subtilis plus Pseudomonas fluorescens. And here you see the phosphate solubilization zone increased significantly as compared to HNB9 alone. So this fungus, this protozoa is now in combination with the fungus is again, enhancing the phosphate solubilization. So we have done some greenhouse results. You can see the difference in the growth, protozoa plus HNB9, HNB9 alone and control. So in wheat and fenugreek. Now, from these consortia, all these three consortium are patented. One of the technology HNB9 alone is already transferred to a company. Now we come to the biocontrol aspect. The biocontrol aspect started with the story of a UG student, Srinivas Patil, who did this work using the culture filtrate of the fungus for the biocontrol of Fusarium oxysporum. And later we also use the HNB9 for the biocontrol of Fusarium oxysporum race 1, which is a very deadly pathogen of banana world over. Right? So what we could see, effective biocontrol, and we pitched a project in ATGC grant, Translational Grant for Commercialization in DBT, and we got a project sanction to demonstrate the biocontrol in 3 crops. So here we are working on subspecies Lycopersicki, Short, and Vasinfectum, and HNB9 plus Enterobacter cloacae is significantly inhibiting the growth of Fusarium oxysporum across these 3 strains of Fusarium oxysporum, as you can see here in this picture, 40%, 40%, and 38.3%. And this is confirmed by Evans blue staining. I'm not going to detail of that. It's basically staining the dead cell, right? And this is plant microbe interaction. These plants, these plants are treated with Fusarium oxysporum, right? And you can see the suppression of plant growth as compared to control not treated. And when we use our treatment, the plant is able to overcome the suppression across these 3 strains. Okay, right? Tomato and okra and fenugreek. So this work is in progress. Now I come to another technology which is transferred to an industry of making a biopesticide. Now this biopesticide is based on 2 entomopathogenic fungi which kills the insects. Metarhizium, Anisoclea, and Beauveria bassiana, they are very well known. But no one in the world has made a consortia of these 2 fungi. Even I pushed the officials of CIB to include the consortia, and thankfully since 2022 they have included consortia in their guidelines. First time they have included the consortia in their guidelines, and they have diverse target insects, Metarhizium and Beauveria bassiana. So what I did interaction study with HNB9 between Beauveria and bassiana, positive interaction. But when I directed Beauveria bassiana and Metarhizium, after 20 days, Beauveria was found to inhibit lyse the cells of Metarhizium. So what we did a tripartite consortium, and this could overcome the inhibition of Beauveria towards Metarhizium. So this was a discovery which we patented. This is confocal microscope imaging, where all these 3 cells, 3 microbes are growing together, all well in the family. And we treated the sugarcane with this consortium, and as reported by the farmer, there was a significant effective biocontrol of white grubs and nematodes. One of our students who is working with me for PhD is now validating its biocontrol activity. but before she could have validated what industry has taken this technology already. And because it is a soil fungi, HNB9, for the foliar application of Bevelia and Metarrhizae, they use a culture filtrate and they could grow together. And also we could use the culture extract, filtrate extract, removing everything from the filter, only using the active ingredient. And we are also right now, analyzing the active ingredient from the DBT project, and will be coming out with its constituent. And again, there is a positive interaction between the two fungi. And we have taken small steps towards now fermenter scale upscale production, and this is in the 3-liter fermenter production which we have done right now. Now, if you see the methods of application, it can be seed. Seed is the best treatment, seed treatment, and we have standardized along different crops. You might be wondering why I could have done so fast, because I am mentored by Professor Dr. Ajit Verma ji, who has already made a very significant discovery of Piriformosphora indica. So we had a lot of field and other experience, our setups, so we could go very fast in multiple crops, checking its efficacy in the farmer's field. Right? So seed application is the best application. Then is the seedling application, seedling dip you can give, or soil application. Right? Now, one of the spin-off, industrial spin-off, is development of an antimicrobial fabric. So this is the fabric, woolen fabric, coated with the extract of this fungus. And not only it is taking up the color, right? But this color, this fabric is antimicrobial. When I say antimicrobial, we got it tested through Sriram Institute of Research in North Campus. It is antimicrobial against pathogenic E. coli and salmonella as per the Japanese standard. So that is it. And even after the wash, giving the wash, the antimicrobial activity is retained. Now my aim is that if the constituents are fit for human consumption, this can be used for artificial coloring, because by 2028, the red color carmine is going to be out. So that is a wonderful opportunity we have. Some recognition we got from the falling wall competition. The tripartite combination was pitched by my student, master student who did this work, and this was selected among the top 15 innovations made by young scientists from India. And then we pitched our technology to grow further, Chicago-based organization, Seattle-based organization rather, sorry. And our technology was selected among top 20 technologies for funding among the 740 applications worldwide. And only 2 were given the funding from the African countries. That's another thing. IFT, Institute of Food Technology, USA. Where there also we pitched, and they recognized our technologies have one of the top 6, centers of cutting edge entrepreneurship around the globe, right? And we were featured in their magazine, and this is what they wrote about us in their magazine. So this is all my work. Thank you. And I would like to acknowledge Department of Science and Technology for the discovery work, DBT for the translational grant work, ICL Israel, they have supported us in the trials. Our Honorable Founder President of MIT, Dr. Ashok K. Chauhan, Chancellor Sir, Dr. Atul Chauhan, and Vice Chancellor Ma'am, Dr. Balvinder Shukla. And this is my team. I am fortunate to be mentored by Professor Dr. Ajit Verma, who is from JNU, and my team, Dr. Swati, Dr. Monica, Dr. Surbi Dabral, my students, Dr. Himesh Joshi, and others are right now all enrolled with me. Chesta and Monica Bisla, who did this consortium development with Bacillus subtilis and Metarrhizium, and Srinivas Patil, who initiated the work on inhibition of Fusarium processurum. So thank you very much once again.
Thank you, sir, for this very translatable work.
Very nice work. Like, so my first question is, why you have not included the S indica, P indica in your concept?
Yes, sir, but they were not talking to each other.
They were not, so what is the whether this your fungus is better than S. indica or P. indica?
I cannot say that, sir, only except for one condition. Uh, in the riverbed condition, this fungus is better. This one, only the riverbed.
Another point that you have talked about the fusarium strain that you have got from banana,
Yes.
but you are checking for the different like
Yeah.
okra and
No. Physical strain we tested only up to the strain level. We are checking with the type strains only, like a persicai or
my last
tomato.
question
Yeah.
is important to for the application and for the usefulness for the farmers. It is also important that it is actually getting survived whenever you are having a formulation like that compound.
Yeah.
So how long it survives?
Sir, it survives. We have data for up to 1 year. It survives.
I mean, what is that?
In
You
the,
cannot
in
have
the, in the
the
vegetable
100% efficiency.
in the vegetable powder formulation,
Right.
when we make the store count of
For
SCLS
bacteria
count,
also I'm asking because
Yeah.
whenever you are having the bacteria and fungus,
yes,
so 1 year is quite like you cannot have
Yeah.
achieve that.
Yeah.
So the 1 year time is quite good. So at least
for
50%
1 year, spore count of initial spore count it goes down, but we are able to keep above the recommended 5 into 10 to power 7 spores per gram of the formulation.
This is true for with the fungus, but I'm talking about
Yeah,
the
with the
bacteria.
bacterium also.
Bacteria also, that you should have a data
Yes,
where the fungus and
because
bacteria
Bacillus subtilis is a spore former. Sir. Bacillus subtilis is a spore former. With fungus plus BS, Bacillus subtilis we have standardized right now, not with other.
So that one has to bring you are
Yeah,
talking about consortium.
Yeah, because Bacillus is a spore former, that is why it is surviving.
Very nice.
One very intriguing, first of all, the very vast protocol. One intriguing findings that the embryophytes will keep in the field.
Yeah,
So it leads to early flowering.
early flowering.
So both promotion can be, we can understand, early flowering is considered as a special sponsor, right?
Yes,
So how does it like, is it that giving
because it is a fungus which is colonizing the root of the plant. So also transmitting the distal responses is stress responses. That is why early flowering.
okay, but will that reflect in the yield?
It enhances the yield, sir. Yield enhancement is there.
Because what we have seen, early flowering mostly are associated with the message. And because they get the shorter time for the accumulate photosynthesis to accumulate. That is what they,
Yeah.
that means in the stress situation, they have to survive,
Yes.
produce the seed.
The flowering is early and prolonged, so yield is more. Sir. What we have observed in the farmer field, we have not done in the lab as such. Yeah.
So microfiber training was done without fixative or the
With fixative, modern fixative. The protocol was given to us by industry, and we followed the normal staining protocol with moderating agent and ammonium chloride, everything. Yeah. Washing. No. Microbial antimicrobial activity was intact even after washing. Yeah. Uh, Not at this stage, but as an antimicrobial fabric, it can be used. Yes, that's a problem because with washing it will go down, but as an antimicrobial fabric, once in before one washing only for one washing only, right? No washing involved, it can be used.
Maybe the medical uses is more there.
Yeah, medical.
Dressing and all.
But if the industry want, they can add some additives and fix it. That's a different thing, right? Because this we have developed. A lot of improvements can be done on the basic dyeing process. So if you can make the dye permanent, right, it will retain its antimicrobial property and also can be used for the daily use because the color is beautiful. Yeah. No, in the formulation, sir was asking, what is the shelf life? Am I right, sir?
every time we need to.
Yes, because we are using it under conventional farming, maybe not in the organic farming, we not require to use it.
So, thank you.
The cesarean infection, you did first and then you treated that.
Yes,
And
yes, yes.
so the morphological symptoms which were because of the infection, they got treated after.
Oh, we infect the plant with fusarium and then we give the treatment of the fungus.
You showed that the plant got itself back and the roots were better.
I have shown you the slide in which there are 5 rows, right? So the control, not treated with anything, then treated with fusarium alone, and then treated with our intervention, right? So after fusarium treatment, one set was treated with our intervention. The next other sets were treated with our intervention. Immediately, immediately. This was simulating the presence of fusarium in the soil. Where you sow the seed, right? Fusarium is present. Okay. Now you sow the seed, fusarium is present, and our.
It would be better to do the 6 o'clock.
Thank you, sir.
Thank you.
Wonderful, sir. Our next speaker is Dr. Mahesh Rao. Sir is a senior scientist at the ICAR National Institute for Plant Biotechnology, Delhi, working on pre-breeding and polyploidy evolution in Brassica. His research focuses on characterizing unadopted Brassica germplasm, land races, diploids, and crop, wild relatives, and transfer of valuable traits from these germplasms or genotypes into cultivated species through wide hybridization. Notably, he developed the wide compatible Brassica rapa variety yellow sarso germplasm, which is important for the wide hybridization program. With a PhD in genetics and plant breedings from BHU and international research experience as a Sir Shore Fellow in Germany, Dr. Rao has made significant contributions to Brassica breeding. His impactful research continues to advance genetic diversity and crop improvement strategy. Sir, I hand over the dais to you. His topic for today is diversifying the primary gene pool of Brassica juncea, leveraging diploid progenitor species and crop wild relatives through a pre-breeding strategy. So, over to you.
Thank you, ma'am, for a nice introduction, and thank you everyone for having me here. And, my apology that I was not available yesterday. So my explanation is today, and I don't think this is the session of the plant breeding, but you will bear with me, and hope you will enjoy at least seeing the slides, and this is more colorful. So my topic is diversifying the primary gene pool of Brassica juncea, leveraging the diploid progenitor species and crop wide varieties through a pre-breeding strategy. So I am Dr. Mahesh Rao. I am working on the pre-breeding Brassica from 2011-12, and I'm continuing this work at ICR and ITB Delhi. So I'm going to give my presentation in following steps. Firstly, we understand the Brassica species and interrelationship between the different diploid and polyploid species, and what is breeding and why it is important. Then what we are doing at ICL Delhi, that is characterization of crop wild relatives, mostly in Brassica. Then we are following 2 parts. One is resynthesizing the Brassica juncea from the diploid progenitor species, and the second one is the introducing the genes and traits from the wild to the cultivated Brassica juncea, that is the anti-diploid Brassica. And then you will see how much lines we have identified and how much stock we have developed and characterized, and then we will draw a conclusion. So firstly, if we talk about the Brassica, we are growing Brassica juncea in India for the oilseed as an oilseed crop, and this year we surpassed the soybean in the production, and now it is ranked 1 in India in all the oilseed crop. And if we talk about the production and the area, the Madhya Pradesh is having the maximum area, covering the maximum area for the Brassica oilseed. And if we talk about the production, the Rajasthan is the number 1 in the production and productivity. So why we are talking about the genetic resistance? So we are trying to move forward towards the attaining the goal of sustainability that we can minimize the role of the chemical treatment for combating the biotic and abiotic stresses. So our aim is to identify the donor lines or traits in the wild species or the unadopted materials, and then we can transfer for cultivated germplasm. So this is the triangular view. If you are from the agriculture or plant breeding background, you might be knowing. So there is three diploid species starting from Rapa, Nigeria, and Indonesia. These are three diploid species having a different genome, different chromosome. And from these diploid species, three amphi-diploid or tetraploid species have been generated in nature. That is the Brassica that is grown in India for the oilseed crop. Then the Brassica carinata and Brassica nepos. Brassica nepos is grown in mostly European countries or China or in the Canada as the oilseed crop. So we will focus on the more on the Brassica. If you talk about the matting system, the most all the three amphi-diploid species or tetraploid species are self-compatible, and all the three diploid species are self-incompatible except the Brassica rapa variety yellow sarso, subspecies the yellow sarso. So, what is the pre-breeding and why we are focusing on that? And we will mostly focus on the Brassica which we grow in India for oilseed crop. So, all the activities to identify the desirable characteristics or genes from the unadopted material that cannot be used directly in the breeding population. So, and also to transfer these traits to an intermediate set of materials that breeders can use for the legal variety of crop improvement program. So our job to develop the a lot of genetic stock, and we can characterize it, and we can provide to the breeders that they can utilize for their varietal development program. And that was very nicely described by the Sivali Sharma et al. In one review paper from Professor Rajiv Vasne group. So they have described how tree breeding work and then how the material can be utilized by the breeders for development of varieties. And it will take time on the basis of species or the crops you are using for the pre-breeding and varietal development program. So we try to depict one picture here. You can see in picture the funnel is having the primary gene pool of the cultivated Brassica. The breeder is opening the tab and they are utilizing this one. But the tab is already not fully filled as the diversity, genetic diversity in the primary gene pool of Brassica is very low, being a qualified crop. What is our job? That we can develop the genetic stock, characterize genetic stock, the pre-breeding approach, and then we can fulfill this funnel that breeder can utilize for their developing the varieties. So, pre-breeding method will lead to enhancing the gene pool in the primary gene pool of Brassica, evolution and domestication of polyploid species we can understand through developing these materials. Then we can go for the development of CMS system development. So we can transfer the cytoplasmic male sterility system from the wild to the cultivated species for the hybrid seed production. Then we can transfer the genes from the wild to the cultivated species for any specific trait for biotic or abiotic stress tolerance. Then we can also go for the development of the new synthetic species, like in case of Brassica, PAU already developed the hexaploid Brassica species. That is the synthetic one that is not available in nature. They have accumulated all the three genome A, B, and C, and they have developed the hexaploid Brassica, and they are doing some research and testing on the production and productivity of that species. Also, we can go for the targeted new plant type development, and then we can what we are doing that we can do the resynthesizing the Brassica yellow polyploid or amphi-diploid species. And why it is important? Because there is very less genetic diversity in the cultivated amphi-diploid species, especially in Brassica Brassica nepos, and Brassica carinata. So due to this limited genetic diversity, there is genetic ceiling for different economic traits. We cannot go beyond more for the yield contributing traits, biotic and abiotic stress tolerance, and also for the CMS system, we have to look forward for the wild species. So what we are doing at ICR and IPV, so we are doing we are characterizing the CWRs of Brassica. Then we are following the two routes. One is the synthesis we are doing from the parental diploid species for the Brassica. Also, one novel strategy we applied for resynthesizing the mutant resynthesized Brassica from the placental species. And also we have got one NRTP Rapa 8, that is the wide compatible Rapa, where we can make the process and we can bypass the embryo rescue protocol while synthesizing the Brassica diploid Brassica. And then we are doing some introduction breeding from wild species. And also we are trying to, whenever we are going to develop the introduction line, there is limitations for the linkage drag. So how we can minimize that? We are trying to do the experiment for that. We can minimize the linkage drag in the advanced information lines. So in the characterization, some of my students is working on that, and these are the wild species we are having at our center and from long back, and we are characterizing it now. Earlier researchers, they have transferred some of the CMS system from wild species to the cultivated for the hybrid seed production. But now we are characterizing for the morphometric traits and also on the basis of morphological and molecular characterization. And this is the distribution of all the wild species we are having at our institute, which is distributed in the different part of the globe. These are some of the study we did on the basis of morphological and molecular analysis. Also, we did some quantitative and quantitative trait we have analyzed. Also, we analyzed the structure of seeds. Also, we are going to see the composition of the fatty acid in the these wild species that we can utilize in our breeding program. So, doing the characterization, firstly, we are going to abiotic stresses, heat and drought. So firstly, we are screening at three different stages. That is the germination, juvenile, and reproductive stage. For germination, juvenile stage, we are doing the experiment in vitro. For the reproductive stage, we are doing the experiment in the field condition. Same for the drought stress, we are doing the experiment in the germination, juvenile, or reproductive stage. So for the germination and juvenile stage, we are taking care of some of the morphological data, and also we are going for some of the biochemical parameters which is associated with either heat or not response, and then we have identified some of the phenotypes which is performing well, which can be used further in the breeding program. So, and for the thought, seedling stage also same pattern we have followed, and we have identified some of the phenotype, some of the species which is working very well in compared to the all the U triangle species. Whenever we are going to analyze the wild species, we are taking care of one representative lines from the all the U triangle species. For the reproductive stage, for the heat screening, we are planning the experiment for the timely and the late soon condition that the flowering can be come in the late February, that it can be hit by the high temperature during the reproductive stage, and then we can analyze the different parameter on morphological as well as biochemical parameter, and then we can accumulate this data, and then we can identify some of the lines. Same way for the drought stress, we are doing with the irrigated and non-irrigated withdraw irrigation system under the Renault system facilities at ICR and IPB, and we have identified some of the genotypes which is working very well in the for the drought stress at the reproductive stage. Same way, one of our colleague, he's a plant pathologist. He's working on identifying resources for the biotic stresses, that is for the powdery mildew, white rust, and we have identified some of the genotypes which is immune to powdery mildew, and that we are trying to transfer in the cultivated species. And all the cultivated species not having the immune or resistance, highly resistant for this pathogen. So we can look for the wild species for transferring the trait. Same way for the white rust. In white rust, there is variation in the different isolates. And so we are targeting that multiple resistance for the multiple isolates we can found, and we can transfer the traits from wild to the cultivated one. So you can see the whatever the lines species we have identified that is tolerant to 10 to 12 isolates, and that we can use in the breeding program to transfer this trait to the cultivated one. Yeah, one of our collaborators from IAI, he's working on Brassica oldacea, and his trait is the Alternaria brassicola that is infecting the Brassica oldacea. So he has screened all the genotypes, and he has some of the genotypes which is coming with the good level of resistance for the Alternaria brassicola. And the same way for the mustard aphid, because it is a very big problem in mustard, and there is no resistance force in the all the cultivated species. They have screened thousands of materials, but they have not found any one here which is giving the resistance against the aphid. But they have identified some of the promising aphid resistance line species that can be helpful for the transferring the trait from the wild to the cultivated one. Same way for the there is one problem of root parasite, that is the urobankin. So we have screened some of the genotype, huge number of genotype of the Brassica gentiana and cultivated species, but still we are lacking of getting any resistance. So what we are thinking, we are trying to get some of the genotypes or species which is resistant to the that is herbicide. So we can apply the herbicide to control this kind of root parasite, at least till we are not getting the any genetic system for the overwhelming. So we have identified some of the tolerant and moderately tolerant lines in the cultivated as well as wild species that can be potential target for our transferring the trait from wild to the cultivated one. So once we have identified some of the genotype which is performing very well, we can go for the wild hybridization program and we can try to transfer these traits from the wild to the cultivated one. So for that we have to do first the crossing, then we can go for the embryo rescue because most of the wild hybridization program we have to go for the embryo rescue. Then we can do the polyviability analysis, we can do the classical as well as molecular cytogenetic analysis. We can go for the ploidy analysis whenever you are making the wild hybrid. Then we can confirm the hybrids. Then we can go for the position treatment for the MPD protection if needed. Then we can go for the bud pollination. Then further certifying and selection we can do, and then we can finally characterize the lines on the basis of molecular, morphological, and cytological analysis. And then we can find out these are the lines which are stable, which are working well for different traits, and then we can utilize in our breeding program. Same way, the first program we have started, that is the resynthesizing Brassica gentium, where we are working with the two different species, Brassica rapa and Brassica nigra. These are the progenitor species. We are making the process and we are doing the amplitude polarization using the position treatment. And then we are developing the advanced synthetic lines. So we have developed 93 synthetic advanced line, which has been identified for the different traits. And also we are doing some of the psychological as well as molecular and stability analysis, and we have identified some of the genotypes which is performing very well for different states, and contrasting genotypes have been identified that will be used for the further molecular biology study. Also, we are doing some of the genomic studies in these lines. So we are doing the SNP genotyping. Also, we are targeting for some resequencing program to see the intergenomic interaction. Whenever two genomes are coming together, how they are behaving towards each other, and how recombination happening between the two genomes. So that we are targeting from even from the first generation to the S8 or S9 generation, 7, 8 generation we have taken, and we are trying to find out the interaction between the different genomes. That work is under progress, and we are trying to analyze the data which we got from the genomic study. Also, we have characterized some early because we have to wait for the 7 to 8 generation for stabilizing that material. So our thought is that we can analyze the genotypes or the material in the early generation, in the second or third generation, then we can directly use for the breeding program for the our target trait. So in the S2 and S3 generations, we have analyzed these populations for the genetic and morphological traits, and we have identified the variability which we are going to utilize some of the genotypes for the different traits in the breeding program. And some of the material has been shared to the different breeders that they can utilize in their crop improvement program. So, and second thing we started doing, we wanted to develop the more diverse synthesized raster and cell lines. So earlier we have to make the process, we have to go for the embryo protocol, and then we can get the F1 plants, and then we can go for the W, and then we can go for developing the synthetic amphibioloid. I missed it. So now we have got one specific RAPA line that we can make the process, we can remove the embryo rescue protocol from our experiment, and then we can get the in vivo seed. And then whenever we get the in vivo seed, amphibioloid seed, then we can go for the experiment treatment, and then we can develop the synthetic amphibioloid, and then we can develop, go for further selfing and selections for developing the different lines. And then we can characterize these lines for the different phases. On the basis of that, we have identified some of the genotypes which are working very well for the different states, for the heat and drought states. Also, we have identified some of the genotypes which are having good level of morphological diversity for different yield contributing traits. So, during our wild hybridization program, we have identified one of the genotypes which is very well for the wild hybridization program. So earlier we have to follow this path, we have to go for the embryo rescue, we have to go to get the synthetic amphibioloid. So now we can make the cross and we can get the in vivo seed whenever we are going to make the wild hybrid, and we can go for the derivation of that seed, and we can follow the further procedure for developing our genotypes. So this is a very important material that we can go for further understanding what kind of genes and what kind of mechanism is working behind this wild compatibility. So one of my students is working on that. and we got funding from the DBT, and we are still working on that. Before that, we were wanted other raster polymer. So you can see in the carbon raster polymer, we are getting 2.7%, 71% in case of raster RAPA A, that we are getting the 35.52%. And in the same way, in the in vivo condition, that is very important, where we are not implying any tissue culture intervention. There we are getting 30%, 30.46% in compared to the 38.38% in the other normal genotype. So employing this, we have developed some of the things. So, so can I get 342? So same way we are developing the information lines, and we have developed some of the synthetic MP deployers, and also one successful example we are having. We developed the Erudapa Junction, that is the advanced information lines, and that we have developed for the alternaria resistance, and resistant lines have been identified, and we did some of pathological analysis to prove that we are working well for the resistance to the alternaria. And this is the one of the methods we used when I was in University of Bonn. We employed the gamma radiation break the chromosomal segment and see the combination how it is going to happen, and that can be employed in the our advanced information line where the linkage drag is available, that we can take the linkage drag or minimize the linkage drag. So these are some genotypes we have identified for different traits, and that we are using for the pre-breeding program. And these are some of the pre-breeding material, genetic stuff we have identified some of the different traits that the breeder can use for further development of varieties. So these are some of the challenges in the pre-breeding which we have to take care whenever we are going to plan for the pre-breeding program in Brassica. So this is the conclusion. And if we talk about at ICR and IPB Delhi, we have developed and characterized a lot of material, genetic stock in the reptile mustard, mostly in the Brassica agencia, that can be further utilized by the breeders for the development of the varieties, and we are implementing that material for studying the intergenomic interactions, nucleocytological interactions. Now, also we are trying to understand the polyploid evolution from diploid to the polyploid species. Some of my students are working on that. So thank you. This is my team, and thank you. So any specific questions, I will be happy to answer.
So that the wild variety that you are all these varieties are possible with the
No, it is not. All the varieties are not possible, but we are testing. We are trying to test with these new lines if there is possibility that we can transfer these. We can develop the synthetic amphi-diploid using the RAPA and wild species. That will be useful for us, that we can transfer this trait from the wild to the cultivated. But of course, there are huge genetic variability in the wild species. So sometimes we are getting seeds, sometimes we are not getting seeds. But for the Brassica nigra, we are getting the seeds every time whenever we are making the process. That is what it is.
ultimately you are targeting for a single breed, or
Yes.
so like you were talking about some of the white rice.
Yeah, for the Brassica agencia, there is two problems. One is the different traits, biotic and
My
abiotic.
next question is why have you got anything for aphids?
No,
Because that is very
no,
important.
because whatever material we have developed through the pre-breeding, all the material have been tested by the one of the entomologists in the IRI. We have not yet get any resistance for the aphid. That's why they are targeting for the wild species. So from last 3 or 4 years, he is screening all the wild species completely, but he is not getting the complete immune. Yes, he is getting some of the low load on the wild species. So we are targeting that wild species. We can transfer the traits to the cultivated.
So let's wind it up, sir. Thank you very much,
Thank
sir.
you. Sorry, I have a lot of time.
Since there is a lot of shortage of time for the validatory function, so we conclude the session over here.
Okay, thank you everyone. Thank you for bearing with this long session. So just a token of felicitation, can we have, can we like request chairpersons to be on dice to felicitate the speakers, please? So can we have the felicitation for Dr. Dhamma Prakash Wankhede? So I would request again chairpersons to felicitate Dr. Prashant. May I request Dr. Felicitate of Dr. Amit? And then Dr. Mahesh Rao, may please. So then finally, as a token of appreciation to the speakers as well as the chairpersons, I would request HOD Dr. Humara Faruqi to felicitate them. So may you please felicitate Professor Amit Verma, Praveen Verma, sorry. So may you please felicitate Dr. Kakoli Dutt.
I would now request all the speakers to come.
So as they were also acting as the chairperson, so they have another felicitations.
I would now request all the speakers to come on the stage to join chairpersons and convener ma'am for the group photograph. I would now request all of you to move towards convention hall number 1 for the valedictory function, and before that, we'll be having a tea break.
So we have a tea break and then followed by a quick validity section.